Hey guys, welcome to our podcast, The Doctor's Dilemma. I'm your co-host, Serena.
And I'm Ellen Kim, and this is our very first episode.
So today we'll be talking about CRISPR, and we learned about it in our biology class yesterday, and it's super cool because it allows for gene modifying.
Yeah, so part of CRISPR is when the protein in CRISPR, Cas9, identifies when like a foreign virus, I think, was it?
Yeah, it came to be when these biologists were looking at cells and a virus, and the only cells that survived the virus were ones that took in a piece of DNA from the virus and put it into their own DNA, and then from there it was able to recognize any other cells with that strand of DNA, and they knew it was the virus, so it protected itself like that.
Yeah, so essentially the bacterial cell saw the foreign virus, when the foreign virus tried to invade those bacterial cells, although a lot of them died off, a few of them did survive, and then it took in the DNA sequence of that foreign virus, which was honestly a really huge discovery in biology.
Yeah, and it led to more discoveries like CRISPR, and CRISPR is made up of two main components, RNA and this protein called Cas9. Mm-hmm. Do you want to explain what that is?
Yeah, so Cas9 basically is the protein in CRISPR, and like we were saying before, it is the part where it recognizes the foreign virus so that when it does come, when the bacterial cells have that DNA sequence of that foreign virus that came in, the Cas9 takes that DNA sequence and recognizes it so that when other new things, it could be other cells or other strands of DNA or actual viruses come, then it can recognize if those DNA sequences match up, those DNA sequences from the viruses, which makes it so they know who it is.
It's like Cas9 takes a strand, a sequence of DNA and puts a wanted poster on it.
Yeah.
And it's every time a new cell or whatever wants to enter this bacteria, then Cas9 will be will search its DNA and see if it has that particular sequence, and if it does, they won't let it enter because they know it's a virus.
Which I feel like is so cool because it's all happens so naturally and isn't adapted or engineered by scientists in a lab.
And it's cool because it came from such it was such a small discovery,
mm-hmm.
and it came to be such a big, and it's super life-changing too because it solves so many cures so many diseases.
For example, sickle cell anemia, which actually yesterday in the documentary where we found out about this, we heard about how sickle cell anemia causes so many people to die in Africa, 5 to 8 children, children ages 5 to 8 years old with this condition die at those ages.
Yeah, and even in the U.S. where healthcare is so advanced, the longest that people live is to their mid-40s.
Which is really crazy.
Yeah. So, currently, if you have sickle cell anemia, then you're, it's very hard to, live past, 40 or 40, 40, what was it? Mid-40s?
Mid-40s, yeah.
So...
Because sickle cell anemia causes the cells not to be full and round, but it to be like a half moon shape, so it doesn't carry enough oxygen. So essentially what people who struggle with sickle cell anemia have to do is go to the hospital every month, every few weeks or so, to get a blood...
blood from a donor?
Yeah, which is really crazy to do because it takes not only a lot of time but a lot of effort,
a lot of energy, and it'll ruin the liveliness of kids. It's really sad. And because we live so close to Sanford Hospital, I've seen patients with sickle cell anemia before. It's really heartbreaking.
Mm-hmm. But what CRISPR does for this specific.
Wait, I can explain. So how sickle cell anemia like happens is when there's one single base in the whole like millions of DNA, but only like one base is changed from it's meant to be an A, but then it ends up being a T through a mutation. And because this one base is changed, the whole sequence is like shifted, and now like and this is how they develop this condition. And just because of this one base. So
This one sugar base, deoxyribose.
yeah. And like it's really hard for scientists to like identify this base and change it back because there's like billions of like strands of DNA within our cells, so it's like impossible to target this specific base with the past technology before CRISPR.
CRISPR is useful because it can change that, or it can identify.
It identifies that strand of DNA and then it
Modifies
it. Yeah.
Yeah.
So this is essentially the very basic information of what CRISPR is. Maybe in the future we'll dig more deep into it in a later episode.
Thank you for listening to our first episode of The Doctor's Dilemma. This was your co-host Serena Ma.
And this is your co-host Ellen Kim.
Thank you.